Greetings, everyone. Thank you for joining another one of our RRP-hosted research webinars. I'm Katrine Erickson. I'm the Executive Director of the Runx1 Research Program. I think I say this every time. I think most of you know who we are and are familiar with RRP, but I always want to make sure that if there's someone new to us, they know a little bit about who we are. So RRP is a nonprofit organization, and we were started by a Runx1 FPD family, and thus we're dedicated to supporting patients with Runx1 familial platelet disorder, which I'm sure many of you on the call know is a genetic disease caused by mutations in Runx1. And unfortunately, this disease is correlated or associated with a significant increased risk of hematologic malignancies, predominantly myeloid malignancies like MDS and AML. RRP, when we were established by the Babbage family, they decided that we would tackle really one primary goal, and that is to prevent hematologic malignancies in Runx1 FPD, and of course also improve overall quality of life in patients. So how do we do this? We do this through significant research funding with that specific goal in mind. But also we spend a lot of time building and supporting our patient community as well as our research community through things like this and our conferences, and of course facilitating collaboration whenever possible across those different groups of really key stakeholders. Our impact in research has now reached over $13 million since our founding, and that has translated into several clinical trials as well. I do want to acknowledge that the $13 million is combined with is inclusive of the dollars that have been spent by our lovely co-funding partners, which include ALSF, the Mark Foundation for Cancer Research, most recently the American Cancer Society, Edward P. Evans Foundation. So we've had great friends in working on this important project or this important goal. For those of you who are conducting Runx1 FPD relevant research, please reach out and let me know if you ever want to participate as a presenter in one of these research webinars. I'd love to hear from you. And then also, if you're a scientist and you're interested in being funded specifically for your Runx1 FPD or Runx1 FPD relevant research, please do look out for our grant programs. We'll be announcing a grant program with the ALSF very soon, which will have a deadline in December. And then we'll have another very large grant program or grant mechanism open soon as well, with a deadline in the first half of 2026. So please do pay attention to our newsletters if you haven't signed up, or come and check out our website, and we have information there. Okay, before I introduce our esteemed speaker today, I do want to just remind you that we'll host a Q&A session at the end, but that doesn't mean that you can't input your questions in the Q&A icon right off the bat. So please do go ahead and submit questions, and I'll have the list in front of me at the very end, and we'll talk through it with Dr. Zon when we get there. And then, of course, if you have comments you want to make, you're more than welcome to share those comments in the chat. All right, without further ado, I'd like to introduce Dr. Leonard Zon, who honestly needs no introduction. He's internationally recognized a scientist in stem cell biology and developmental biology, in cancer genetics. Dr. Zon is the Grausbeck, I hope I said that right, professor of pediatric medicine at Harvard Medical School. He's an investigator with the Howard Hughes Medical Institute and the director of the stem cell program at Boston Children's Hospital. Very fortunately for RRP, Dr. Zon is also a RRP Scientific Advisory Board member, and he's really been a key advisor since the very beginning, for which we're all very grateful for. Today, Dr. Zon is going to tell us about one of his latest research projects, which is now available on bioRxiv if you haven't read it yet, and that is entitled Hematopoietic Stem Cell Division is Governed by Distinct Runx1 Binding Partners. We're delighted to have you here with us, Len, and I really look forward to your presentation.
Well, thanks, Katrine. Super happy to be here, and I have great, pride of being part of the Runx1 family. So I'm going to talk about three stories, actually. One of them is based on my title, but just so we set ourselves in the right pace here, you have hematopoietic stem cells that can self-renew and can differentiate to the blood lineages. And these are facilitated by niche cells, which include the vascular endothelial cells and the stromal cells in the marrow. Stem cells are one of the only stem cell types that have a quantitative assay, and stem cells are used therapeutically for transplants. I don't have to tell this audience that. And when mutations occur, you get clonal expansion, and this can lead to leukemia. So several years ago, Owen Tamplin in my lab made a transgenic fish that had a green stem cells. And he used the Runx1 enhancer that drives green fluorescent protein. And then when we looked at the colonization of the niche, which is here, this caudal hematopoietic tissue, we could see that the stem cell arrives and then it goes outside the blood vessels. But then, strangely, the blood vessels wrap back around the stem cell. We called this endothelial cuddling. Then there's a division, and one stem cell leaves and the other one stays. And this is a very stereotypic behavior. So we're very interested in how the niche interacts with the stem cells and what are the programs inside the stem cells that are driving this fate. Now, in my lab, we've been studying stem cells for a long time, and one assay that we use to study them is transplantation. So in transplant, you can look at clones of cells, but it's a little bit difficult because you usually irradiate the host, so the host niche is inflamed. When you take the stem cells out of the body, the transplanted cells are stressed, and only some clones contribute after the transplant. So in my lab, we've been working on some barcoding technologies, in vivo barcoding technologies. We have two of them, one where we color every stem cell a different color, and the other where we use a CRISPR barcode system, and these are inducible, and you can simply bleed the animal or do a marrow analysis, and there's no transplant, there's no stress, the stem cells are in their native state, and they're an accurate assessment of clonality. So throughout my talk, I'm going to be discussing all of these technologies. Well, several years ago, we published a paper where we could take zebrafish embryos and disrupt them and basically do a blastomere culture assay. And originally, when we did this, we found that a lot of the embryonic blastomeres make muscle. And we published a paper in Cell in 2013 where we were able to find pathways that induce muscle. This was by adding chemicals to the media and just culturing the blastomeres and looking for the induction of muscle. Well, once we did that, we decided we have these beautiful Runx1 fish that I showed you that are, when the stem cells there, they fluoresce green. Why not try to do a chemical screen in very high throughput to see what would induce Runx1? And we screened through 3,800 chemicals in duplicate, and we identified 21 positives. But probably our strongest hit was strangely an inhibitor rather, of Runx1 CBF binding. And you can see here, you see all these little green dots here. They're all the Runx1 positive cells induced by this compound RO5-3335. I'll refer to it as RO5 for the rest of the talk. So RO5 was originally found by Paul Liu. And this was a massive screen, biochemical screen. And what he was trying to find was a compound that disrupted the Runx1 CBF beta interaction. Now, it was unclear that this compound actually did this because others in the field, and I'll describe that in a second, thought that this compound looked more like an inhibitor of bromodomains rather than a direct inhibitor that would block this interface. And suffice to say that the molecular target for this is really not so well known, but it suggested Runx1 inhibition. When they gave it to zebrafish embryos, it blocked the production of, blocked the birth of blood stem cells. And this greatly phenocopied a mutant in the field of CBF beta mutant that lacks definitive hematopoiesis. So everything in the early embryos suggested that this was involved in blocking Runx function as it starts to bud off blood stem cells from the aorta. Well, Ann Ellett in the lab was very interested in studying this compound because it came through in this screen that we had done. And we added the chemical at 24 hours up to 54 hours. And this is a little bit later than adding it from the very beginning of embryogenesis. And what we clearly saw was there was a tremendous increase in the number of Runx1 positive cells in vivo in the embryo. And you can see that here, tremendous increase in the number of cells. So we wondered whether this was involved in hematopoietic stem cell specification, or was it involved in stem cell division? So one of the things that you can do is look at the number of budding events from the aorta where the stem cells are made. And again, we're adding it a little bit late here, and we don't see any effect of the birth of the blood stem cells. So I think if you add this earlier in development, Paul showed that you'll suppress blood stem cell production in the aorta, but not when you do it the way that we're doing it. So then, because we could do such fine imaging, we were able to image. And normally when we're looking over a 12-hour period, let's say, you get a few divisions of stem cells. But now when you do RO5, you get all these beautiful divisions that happen. And you get many more divisions. So the way to think about this is probably the stem cells are born in the aorta. They travel to their new site. As they're traveling, they execute normally one division. I think what's going on here is this chemical is inducing at least a second and maybe even a third division as the cells are actually hitting the niche. And you can see that here by the number of Runx1 positive cells in that region. So, we thought this was interesting. It's one of the few chemicals that can induce symmetric divisions of blood stem cells. And we wondered, how does this affect the downstream pathways, downstream lineages? So in this experiment, what Anne did was to have a fish that had fluorescent macrophages. And she added this for 7 days in a row. And what this did was actually suppressed macrophage production. And you can see that here. Now, if you just add it till the 54-hour point, and again, that 54-hour point is exactly when that division is going to happen. And then wash it out. Okay, now you get more macrophages. So what this suggests is a model where maybe this compound blocks differentiation, thereby increasing the number of symmetrical divisions. So again here, with RO5, if you add it later, you're going to suppress differentiation, but adding it earlier, you get more symmetrical cell divisions. Okay, so what about clonality? So we published this paper on using the zebrafish system, and we were able to find that a typical zebrafish embryo would have 20 to 30 blood stem cell clones. And irradiation or transplant reduces the clonal diversity. Now, we also published a paper recently about macrophages removing blood stem cells through an eat me signal, CALR, but macrophages that have beta-2M are allowed to proliferate. And then we also showed in a science paper that Runx1 mutant embryos have fewer stem cell clones that contribute to adulthood. So we wanted to see what happens to clonality with this compound. And I have to say, this is a real amazing result. we used our ZebraBow technology and we added the chemical plus tamoxifen and then measured the number of stem cell clones in adulthood. And you can see here really nicely the number of clones from a normal induction without the drug, but you get many more colors if you've given the drug. And when we look at the hematopoietic stem cell clones, you get a substantial increase in the number of stem cell clones. And you can see here on the cluster size that you also have an effect. And so what happens is that you get many smaller hematopoietic stem cell clones with an increased clonal diversity. So that's very exciting. So this compound is really special in its mechanism. So we wanted to see what happens with this RO5 compound. And we actually took Runx1 GFP, which are also tagged with a ubiquitous M. cherry, and then transplanted the cells into embryos, and then waited till adulthood to assess the percent of engraftment. And you can see that RO5 leads to a substantial increase in engraftment. And yet there's no lineage bias here at all with this. So they're normal lineage output, but you get much more engraftment. So what we've shown here is that RO5 expands stem cells in the zebrafish. When we look at transplant assays, we see enhanced engraftment. And we also see that the diversity that clones is more with the RO5 treatment. And so this made us really wonder whether this pathway was conserved in any way with mammalian systems. And we also wanted to understand the mechanism of action. So we now switch to CD34 cells. With RO5, you get a substantial increase in the percent of CD34 cells in live cells. And when we do this and look at CD90 as a more enriched marker for stem cells, you get a substantial increase in the stem cells, but you also seem to block the MPPs. And so we think this is a very significant stem cell effect. And again, I think one of the few compounds that truly gives you an extra round of replication. So when we look again at this particular CD90 gated cells, we could also look at EDU labeling. And so with the EDU labeling, you can get more, again, cycling of the stem cells with the RO5 compound. And again, you get this in that CD90 population in the stem cells. And again, there's a decrease in the MPPs. So this is sufficient to say that RO5 expands blood stem cells and probably prevents their differentiation. So how does this do it? Well, again, we had that paper from Paul Liu. And then we have this paper from the Bushweller lab, which showed basically that it doesn't probably affect the interaction of Runx1 and CBF-beta, but there was not really a great conclusion here. But we worked with John's lab to add some of his bromodomain inhibitors that would affect Runx. And what you can see here is these just don't do anything compared to RO5. So there's something special about RO5 and its ability to affect Runx1. So with all of that, we decided to do ChIP-seq on the human CD34 cells. And this was the work of Avik Chowdhury. And the first thing that happened, again, this is CD34 cells treated with DMSO or RO5 for 24 hours. And the first thing that happened, which was a big surprise to me, is actually RO5 increases the binding of Runx to chromatin. So that was a surprise because based on the proposed mechanism of inhibition of Runx, you wouldn't have thought there'd be increased binding. Now, a second thing we did was ChIP-seq for CBF-beta. Now, the chemical was supposed to block the interaction between these two transcription factors, but we don't see any change at all in CBF-beta binding. And so this increase in Runx binding was interesting, and made us try to think about what are the other partners that could be interacting with Runx1? Are there cofactors? So again, what we decided to do was motif analysis, looking at what's underneath the Runx peaks that have the increased binding after RO5. And what we found was that ELF transcription factors, binding motifs, were enriched in these increased peaks. So that made us wonder about ELF1 binding. And so we went ahead and did ELF1 ChIP-seq. And you can see, again, a tremendous increase in the amount of binding of ELF1 adjacent to where Runx1 is also increased. We went on to do communoprecipitation, demonstrating that with an anti-Runx antibody, you would actually pull down ELF1. So we wanted to understand about the genes that were bound between ELF1 and with Runx, or Runx1 by itself, or Runx1 with ELF1. And we saw cell cycle genes were particularly regulated. And because we saw symmetrical divisions, it made a lot of sense for us to study this. So what we found is a number of cell cycle genes like cyclin D2 and cyclin D3 that were bound by Runx, and with RO5, there was induced ELF1 expression binding to those loci. And we also saw some other genes like IL-8 and IL-7 receptor, for which this also occurred. So our thought is that there's cell cycle gene control by Runx1. This has been known by Nancy Speck, Paul Liu, and others, but that there would be a change in the cell cycle genes based on the ELF binding, and this would allow you to go around the cycle and basically do an extra round of replication for your stem cells. So we are working on studying ELF1, but we have ELF2 as one of the genes that we've knocked down with the morpholino. This suppresses the effect of RO5. And we've also looked at clonality, and this also suppresses the clonality that we see. And so this is very exciting functional information that you need ELF factors to basically have Runx1 work for its cell cycle genes. So a summary of this would be that we have RO5 increases the diversity of clones, increases engraftment, affects differentiation depending on the amount of time you add the chemical. Without RO5, you have Runx1 and CBF-beta hanging out probably on the cell cycle genes. But now when you get ELF binding to this, you really activate those cell cycle genes, and we think go around a cycle. And this leads to the stimulation of that pathway. So what I'd like to do is to now extend this with a brand new talk. I should say all the data that I'm showing is completely unpublished. But I'd like to talk about the role of cis-LTR1 in hematopoietic stem cell expansion and crosstalk. And this is the work of Lulu Yu, who followed up some of the work on RO5. So again, I showed you the endothelial cuddling here. But one of the things that Owen had shown in his 2015 paper was that there are 5 endothelial cells that surround each stem cell. And the stem cell is physically attached to a stromal cell, and the divisions are usually on top of that stromal cell. So I already showed you the data about RO5. And the nice part about RO5, although it came from a screen that was an in vitro blastomere culture assay, it taught us actually that this 54-hour time point is quite critical. And if we were able to do a chemical screen at 54 hours, we would find other pathways that would upregulate the division of stem cells. So Lulu executed a screen where she added chemicals, and now this time looked at 54 hours and looked at the division of stem cells. And one of the compounds that came out of this was this LY compound, which is a cis-LTR antagonist. So this was very exciting. We got a tremendous increase in the number of Runx1 positive cells. So she went on to use a morpholino to cis-LTR1 and also to make a germline mutant in cis-LTR1. And in both cases, you can see the increase in the number of Runx1 positive cells. Now, cis-LTR1 is a G-coupled receptor and binds to a lipid, leukotriene D4. And when we inject into the circulation LTD4, we actually suppress stem cell division. So Lulu is interested in studying the expression of cis-LTR1, and you can see here it's expressed in the vascular cells that are present adjacent to the stem cells. And when we look by single-cell RNA-seq of the tail of the fish over here, you can see that very significant numbers of venous sinusoids actually express cis-LTR1. When we look at adult animals, we can see cis-LTR1 is also expressed in these endothelial cells. And you can see here's immunostaining for those venous sinusoids. So when we give the inhibitor, we get many more stem cell divisions. And you can see here in the circles how these stem cells are dividing. And they divide in a very characteristic way. But again, you can see the number of divisions are very significant. And here you can see the divisions kind of where one stem cell leaves and the other one stays. Again, this very characteristic division that occurs in that pocket that's generated from the endothelial cells. And so Lulu looked to see at the pocket, and you can see again, you get more stem cell divisions in the pocket. So we think that this might be part of the cuddling mechanism. So those endothelial cells remodel and make this structure of a pocket. The pocket probably has the ability to sequester growth factors, so they may act at a higher effective concentration. So Lulu was able to take embryos that have venous sinusoids marked in GFP and add the chemicals and then do RNA-seq to see what genes are turned on or turned off. And what she found was a series of genes that affect endothelial function, in other words, how tight the junctions are or not, but also she found interleukin 11. And so interleukin 11 is induced when you give the cis-LTR antagonist, and it's blocked when you give the LTD4. Okay, and you can see here IL-11. So this set up a model where we would have LTD4 activating the cis-LTR receptor on the endothelial cells. This would make IL-11 be produced probably at a very high concentration of the protein in that pocket, and we would think this would lead to stem cell production. So what Lulu did was to inject IL-11 mRNA into embryos and see whether this would rescue the LTD4 effect. And so shown here, you can see LTD4 will lower the number of stem cells, but if you give IL-11, you get a large number of stem cell increase. And then if you block this, you still get that increase if you've given LTD4. So this rescues the LTD4 suppression of stem cells. And so we think IL-11 is one of the key targets of this pathway. So our model currently is that we have cis-LTR inhibition. This leads to IL-11 production and also some changes in angiogenesis and vascular integrity. This pocket is either loose or tight, thereby allowing growth factors to be in the pocket and effectively controlling stem cell division in the pocket. And so it opens up a whole new avenue of looking at endothelial cuddling. Okay, I'm going to end with a story about leukemia, about AML. we were able, this is the work of Chloe Barron, and we overexpressed MYC in blood cells. And for the first time in 25 years of trying to get a leukemia model, we finally got a leukemia model that's very robust in the zebrafish. And this leukemia is transplantable. So it is important that we're overexpressing MYC here, and I'll come back to that at the end. So we've been using our barcoding system, and this is the Gestalt fish. So what happens here is we inject Cas9 and a guide RNAs 1 through 4 into the one-cell embryo. This labels the first 4 positions of the single array in the genome of the fish. Every organ has a different barcode now. And then later at 28 hours, when the stem cells are born, we heat up the fish, and this induces Cas9, and we get these positions now labeled. So this is very useful for understanding the leukemia. But in this fish, it's a cool fish. It's got endothelial cells that are green and red stromal cells. So we're able to also track the barcodes in the niche, and I'll get to that in a second. So first of all, kind of a boring result. Each leukemia is one clone. Here you can see in color-based system, 1 or 2 clones. So, cancer comes from one clone. That's what this shows. Now, the surprise that we had is that the endothelial cells of the niche are oligoclonal. So are the stromal cells. Now, there's an old hypothesis that if a patient has leukemia, the leukemia changes the niche. The niche then is better supportive of the growth of the leukemia. And we favor that type of a model. So shown here is single-cell RNA-seq of the healthy marrow and the leukemic marrow. What you can see here is in normal animals, they have angiogenic progenitors that will differentiate to the venous sinusoids. Now, in the leukemic marrow, what we see is this massive increase in those angiogenic progenitors. We think the leukemia is addicted to the angiogenic progenitors. So how could this happen? Well, what we can see here is the expanded endothelial clones. There are dominant clones of endothelial cells that happen in the leukemia. And there's also dominant effects on the stromal cells. So one way this could occur is where a growth factor is made by the leukemia, and that growth factor activates receptors on the endothelial cells to change their fate and clonally expand a particular type of endothelial cell. This would set up a loop to keep the leukemia growing. So we found a gene called apelin, very highly expressed in the leukemic progenitors. The apelin receptor is expressed in these progenitor populations, but you can see in the leukemia there's a tremendous expansion of the apelin receptor positive cells. So this was a good candidate. We have, there's 2 ligands for apelin. Other people have made apelin double mutants, and they have angiogenesis problems. Ralph Adams has published really nice work showing apelin-positive endothelial cells are necessary for transplantation and vascular regeneration in the marrow. So these endothelial cells that have the receptor also can express the ligand. So there's a little autocrine loop for the endothelial cells, but we think also the leukemias are secreting a lot of apelin. So the way we decided to prove this is we overexpressed apelin on normal stem cells and redid the analysis. So the first thing we saw was there was dominant clones of endothelial cells. And so that was very exciting. This looks very similar to what we saw in the leukemia. Now, a surprise for us is when we measured the blood clonality, remember we're overexpressing apelin in normal stem cells, but we start to see that the myeloid cells have dominant clones. So this is almost like starting clonal hematopoiesis, and you get these dominant clones. So in the literature, there's been some examples where the niche can influence the actual origin of MDS or leukemia. So Dave Scadden knocked out the Dicer mutation, knocked out Dicer, I should say, and got AML or MDS. You can knock out APC, which is part of the Wnt pathway. A number of labs have done this and gotten AML. And Stu Orkin knocked out RB and got MDS. And all these knockouts were just in the niche, in the stromal cells. So none of these have been growth factors, but our work now starts to hint that this growth factor pathway is really required for this interaction. So when we look at the apelin overexpressing marrows compared to the healthy marrows, you get an increase in the number of angiogenic progenitors, just like what you saw in the leukemia. And this is clonal growth. So our model would be that the leukemia is secreting a lot of apelin. Then this leads to dominant clones of endothelial cells, and these clones then facilitate the growth of the leukemia, giving you this loop that is present. And, you know, one of the things that we know in humans is we haven't changed the way we've treated leukemia in 45 years since I was a fellow. We give 3 and 7 to young patients. We now give venetoclax and azacitidine, but that's for elderly patients usually. So, you know, this is a very interesting avenue. So if we were able to inhibit apelin, we think there'd be a great impact on leukemia. So we made apelin mutant fish. We gave them leukemia. The wild type animals get leukemia, but even the heterozygotes are protected from their leukemia. So I think of leukemia as an autonomous mutation inside the stem cell, but I think this niche interaction has been ignored therapeutically, and it's possible that by altering the niche, you could actually modify how the leukemia grows. Okay, what about apelin and AML? So almost all AMLs have high apelin levels. And you can see this in staining based on a few samples that we've done, and we're doing more. So we particularly focused initially on the beat AML data. And, you know, we have all these leukemias, but we wondered what are the ones that are most expressing apelin? And so we subsetted this data. And you can see healthy cells here, but how most leukemias express a lot of apelin. But this particular leukemia, which is inversion 16, CBF beta leukemias, give very high levels. And I should say the Runx1 levels, AML-EDO ones also have high levels, but these are the highest. So schematically put here, you can see here this very high levels of apelin. So we wanted to replicate this, and we have a nice collaboration with Charles Mulligan from St. Jude's, and he provided us with this data, which looks amazingly similar to what we had from the beat AML data. You can see here CBF beta leukemias have very high levels of apelin. All these are actually high, I should say, but these are the highest. And then we talked to the Andreef lab at MD Anderson. They have very high levels of apelin also in this CBF beta one. And then Guy Savageau also showed this, very high levels in AML. Interestingly, there's also some pretty high levels in T-cell ALL. And again, when he subsets to the inversion 16, you get pretty high levels. So as we said, the beat AML data has been replicated in 3 different data sets with literally hundreds of patients. So we're very excited. Through the Runx1 Foundation, I've gotten to know Lucio Castilla very well, and he has a beautiful set of papers on CBF beta fusions with Paul Liu and on his own. And you can see that these animals do get a leukemia. Now, he has a cell line that he made with this CBF beta fusion, and he's given an inhibitor that blocks the interaction here between the CBF beta fusion protein and Runx1. And this affects the transplantability of this particular tumor. We were able to look at appellant levels, and these cell lines, this cell line has very high appellant levels. And then if he gives out this, this inhibitor, it reduces it down. And we know that this is dependent on MYC. And remember, our tumors are MYC driven, and so are the CBF beta leukemias. And so probably MYC is activating appellant. And he was also able to block the fusion protein with an siRNA, and this also reduced the level of appellant. So we think that this CBF beta malignancy is a very good in vivo model in humans of our zebrafish. So just last slide is to show that, you know, we think that in most leukemias will secrete amount of appellant. You also have this autocrine loop of appellant being made by the appellant receptor positive cells. And so this whole feed forward loop is driving the leukemia. And we'd love to be able to inhibit appellant to block the leukemia. But I'll also point out that in MDS and clonal hematopoiesis, so far in some preliminary studies, we've tried to see whether the clone of stem cells is secreting appellant. So far not, but we see very much expanded in mice with clonal hematopoiesis, appellant receptor positive cells. And so we think this autocrine loop here is going on at the beginning of the MDS and clonal hematopoiesis, and then transitions when the leukemia starts to secrete a lot of appellant. So that's my talk, and this is my laboratory. I talked about Ann Robertson, now Ann Ellett, Lulu Yu, and Chloe Barron's work, and the rest have been very helpful, and some collaborators. And thank you very much, and happy to take questions.
Thanks so much, Len. Always an exciting, interesting presentation from you. We have a few questions, but those of you who might have questions in your brain, please put them down in the Q&A icon and we can have a discussion. So the first question / comment. Hi, Len, very interesting, from Nancy Speck. Does RO5 directly interact with ELF1?
Yeah, so we don't really know. My bet is that based on Paul's work, that there was an interaction with Runx1. So I think it could be affecting how Runx1 bends and actually interacts with ELF1, but we haven't directly shown that. So, you know, this is future work.
Great. And then another RO5 question. Great talk, Len, from Wenbin. You might have shown this, but did you have data confirming that RO5 effect is, similar question, is via Runx1, and how RO5 promotes Runx1 DNA binding together with ELF1?
Yeah, it's really the exact same question. So we haven't in vitro done some binding assays with Runx1 and then mixed in ELF1 to see. Those are good experiments. We should do those. Absolutely.
Absolutely. I was curious, you know, as you think about, well, one, you mentioned that this is one of the only compounds you know of that it can actually induce symmetrical division of HSCs. I didn't even know there were others, but I'd love to hear what those others are. But then secondarily, what do you think are the low-hanging fruit, you know, in terms of how to use this therapeutically?
Yeah, I mean, we've talked about stem cell expansion as one important avenue. Obviously, the more stem cells you transplant, the better off you are. And so I think that that would be one use. We've actually been, since we put the paper on bioRxiv, a few other people have tried RO5 and actually gotten a similar result. So I think it's, you know, it is pretty unique. When we finally did our chemical screen, when Lulu did the chemical screen, she also got SR1, which is one of the compounds known to induce stem cells. And so that's another molecule that probably has this type of activity. So, but I don't think there are very many of them. And so, you know, and particularly this one, which likely works at a chromatin level, you know, I think is pretty fascinating.
Yeah, I mean, I could see it also being used successfully in the future for optimizing gene editing protocols, right?
Exactly, exactly. The thing that I was really interested in, it's very theoretical, is clonal diversity. So, you know, I think we're all born with a certain number of stem cells. In mice, some mice have tenfold less stem cells than another mouse, and yet they're still fine. But you probably have a different response to stress. And so if you had RO5 or something like that, you might be able to respond to stress better because it's increasing the diversity of clones. And I wonder about, people who have aplastic anemia, and then you need to bump up their clonal numbers, things like that. So there could be some uses for it in that level.
For sure. Okay, from Alan Cantor, does RO5 affect interaction between Runx1 and other ETS family transcription factors?
Yeah, we haven't studied the others. Alan, it would be great to collaborate and do that, actually. We focus most on the elf family since that elf motif came up. We do know, I think very likely, that elf1 and elf2 are affected. So maybe other ETS family members could do it, but we haven't tried it.
And then another question, everyone's interested in RO5.
Great.
Why RO5, can you comment why RO5 is similar to bromodomain inhibitors?
Yeah, the only thing was that structurally, it looked like some bromodomain inhibitors. So it was based on the chemical structure. So there was a theory that because it looked similar to some other bromodomains, it would be a bromodomain inhibitor. But I think our work that Anne did, where we added chemicals of that John gave us, and they didn't work, they didn't do the same thing as RO5, suggests that RO5 is relatively unique.
Different, yeah. I have a question about the CD34s you did all the experiments in. Are those stem cells from young, like pediatric individuals or older? And
I'm sure they're from older patients. We get them from Seattle, and they're donors who donate CD34 cells, and so we have a diversity set of CD34 cells that work really well, and so they are mobilized peripheral blood CD34 cells.
got it. And you're not seeing any sort of spectrum in terms of the response of 34s to the inhibitor?
No, it seems it's
Pretty
multiple
consistent.
donors, so I think it's pretty consistent. And as I said, what's interesting is even some of our friends have told us the mouse it works and other things. So I think that it's probably pretty robust across species and donors.
Really interesting. Okay, next question. Do you have any hypotheses for other molecules like apelin, which may be involved in selection advantages between hematopoietic stem cells and the endothelium, and how the relationship may be similar or different in clonal hematopoiesis without dysplasia, cytopenia than frank leukemia?
Yeah. So we really don't know any other one from appellant. We tried two other growth factors. As you might expect, the amount of work to do this it requires a sizable effort. One of the ones that I thought had a shot was VEGF. And,
Mhm.
VEGF is a strong angiogenic factor. And we thought if you give VEGF, you would clonally induce changes in the endothelial cells. So we overexpressed VEGF, and we barely saw any change at all, really, in clonality. So apelin is pretty unique. I should have said apelin is a 13 amino acid peptide that binds to that G-coupled receptor. So it's pretty unique. But it wouldn't surprise me that there are others. There's another ligand called apella, and in patients with CLL, there are high levels in the serum of apella. So there could be versions of this. And then the other is, there's probably per malignancy or per type of cell differences in growth factors that are made. So this is an example of how it could work. I don't know if it's completely universal, although, as I said, most AMLs have a fair amount of apelin. In the MDS, we've been working with Jen Trowbridge on some of her models, and we see changes in the apelin receptor positive endothelial cells in the clonal hematopoietic situation. And I think that that would indicate to me that that the niche effects are actually happening all the time in clonal hematopoiesis, MDS, and in leukemia. I think it just gets worse and worse or more solidified as a cycle as you head towards the leukemic marrow. One of the things that also true is we don't understand the effects on the stromal cells. And that's a whole other project, to be fair, but to try to understand stromal cell clonality and what's driving this process. So those are some other future experiments.
Yeah, absolutely. Another question from Nancy. I'm trying to get my head around how RO5 is increasing clonal diversity. If an HSC normally divides once in the caudal region of the fish, but now divides 2 to 3 times, do each of those daughters represent a new clone? This is a really great question because I too was wondering, once they emerge from the AGM, isn't that basically your set standard of clones? And now
Yeah.
you just, yeah.
Yeah. So yes. So I think what's going on is at the very beginning, after you have budded, you have more divisions in circulation as the labeling is going on. So shortly after AGM, you get more clonal diversity as a result of that. But Nancy is correct. If you have a single stem cell and it divides, it would be the same color.
Clone, right.
Right? But if you're at the beginning when the labeling is happening, then you get more colors.
You're at the beginning when the labeling is happening, you get more colors.
So
But does that mean that's an artifact then of like, is it still truly clonal diversity if they've
I
emerged
think
from the
it is.
AGM?
I think it is because they already have budded. So they're already getting ready to make cells. So they're a bunch of cells that can make stem cells. Let me be more explicit.
Okay.
There are a bunch of cells that can be stem cells. What RO5 is doing is putting more of them into cycles so they are stem cells.
Okay. Okay. That makes sense.
But my theory is this happens very early because I think if you're watching a certain colored stem cell divide and it makes the same color, you won't increase the diversity.
Got it. I have another question about your cis-LTR1
Yeah.
part of the presentation. I was curious, I think you were only describing experiments in fish and did you show anything in mice? Anything, what's happening in humans in the venous, in the
So
sinusoid venous cavities?
we've used the antibody to cis-LTR to stain human marrows. This is about all we see. So it stains a subset of venous sinusoids. And we think those are the ones that are the most cuddling, but we don't have any functional data in the human endothelial cells. We've started to grow venous sinusoids from iPS cells. And so we're investigating now about cis-LTR and what expression level it is. So we're just staining some of those cells and seeing if we can then model this in vitro with human cells.
Great. So do we know that cuddling happens in the human bone marrow or not yet? Do we know that at least that process of
We don't know that cuddling directly happens in a bone marrow, but based on Sean Morrison's work and the work of others too, that the stem cells are really adjacent to the venous sinusoids. We know that this happens in mice and in fish.
okay.
And even in the adult, Owen has some beautiful papers on the adult niche having these cuddling endothelial cells in the marrow. And he had also shown in the mouse that this works also. So we haven't, it's kind of hard to image human marrow.
Yeah.
But if you want to volunteer, we could try.
Oh. All right. Any other questions from the audience? There's a lot to digest here, Len, as always. It was a great presentation. I'll
Okay.
give folks a few more seconds to ask any burning last questions. Oh, there's a chat here somewhere. Oh, that's just please submit questions. All right.
That's awesome.
Well,
Well,
if there's no other questions, thank you again for your time. And thank you everyone for participating and for the great questions and have a great rest of your day.
thank you.
All right. Take care. Bye-bye.
Bye-bye.